Hello people, I'm Dr. Rohanmouli and I hope you guys have been through my introductory video, and according to that we'll be starting off with the general topics first and then we'll moving on further with the systems. Okay, so to begin with the general topics we'll have to go through embryology and then the other topics. Okay, so starting we are going with embryology. Okay. So in embryology, before doing anything, all and everything that you need to know is about chromosome, the gametogenesis and the formation of pharyngeal arches. That is all and everything you need to know in general embryology. The systemic embryology, like for each system, what is the development of the heart, lung, or let's take brain, all these things I'll be dealing with in the systemic topics. Here we are learning only the topics that is required in general embryology. Okay, so without wasting any further ado, we'll go with the general topics of embryology. So the structure of chromosome is very important to know because it has two arms, that is P arm then the Q arm. Okay, so in the P arm there is something known as SRY gene. So SRY gene is the gene which decides which gonad the baby is gonna have. Okay, so SRY gene is the gene that decides which gonad it is gonna have. Okay, so this SRY gene is in the P arm of the chromosome. That is all and everything you need to know about the chromosome. Okay, don't waste any time learning about any other things about the chromosome. That is all you need to know within few hours of you going into the exams. SRY gene is in the P arm of the chromosome. Then coming to gametogenesis, we have the steps of oogenesis and spermatogenesis. Oogenesis, which happens in the female, and spermatogenesis, which happens in the male. But here there are two steps that you need to remember in oogenesis. Okay, so that is all and everything you need to know because this is what they have focused in all the competitive exams. The reason why this is very important, I'm gonna tell you. Okay, so during oogenesis, the oocyte will get arrested in two different steps. Okay, so those steps are prophase 1 and metaphase 2. Okay, so what happens is during oogenesis, in the prophase 1, the oocyte will be arrested in diplotin stage. Okay, prophase 1 until puberty, it will be arrested in diplotin stage. Once the puberty is crossed, like once the female genite, so it attains puberty, what happens is it will it will pass the arrest and it will go on further division of meiosis. Here see, it is undergoing meiosis. Elsewhere in the body it is undergoing mitosis. Only in the gonads is where we can see meiosis. Meiosis is 2n to n, whereas mitosis is 2n to 2n. Okay, so here it is the division of 46 chromosome into 23 of the mother, and in father it is going to be again 46 to 23 because during embryogenesis, during the fertilization, these two things will combine and form the 46 chromosome. So that is why it is going to be meiosis in the gametogenesis. Okay, but only thing, forget everything that I said you, only thing that you need to remember and the questions that they were focusing on is the arrest stages. Okay, so in oogenesis, the first stage of arrest will be in the prophase 1, which is specifically they have asked diplotin stage. See, in prophase 1 also there is many stages, but it is gonna be diplotin stage that it's gonna get arrested in until puberty. Okay, this is prophase 1. First arrest, prophase 1. Second arrest will be in the prophase 2. 1 for 1, 2 for 2. Okay, see, in meiosis, you know that prophase, metaphase, anaphase and telophase are the 4 phases, right? So prophase 1 will be the first stage. So remember it as 1, 1, 1. First arrest, prophase is the first thing, and prophase 1 is the arrest that it's gonna take place in. Then for second arrest, it's gonna be 2, 2, 2. That is second arrest in the metaphase 2, which is the second stage of meiosis, and it is in the metaphase 2 that it is gonna take the arrest of. Okay, that is all and everything you need to know in the gametogenesis or the oogenesis, to be precise. Then coming to spermatogenesis, not much of importance has been given here. Only the importance was given after spermatogonia, type A and type B will be formed. That is the only question that was asked in a few years back. That is the only question that was asked with regards to spermatogenesis. Okay, all you need to remember with regards to gametogenesis is the arrest and the step that the spermatogenesis is gonna carry after the spermatogonia formation. Okay, that's it. That's all. Then moving on further, now that you know that the gametes are formed, so now the gametes will be fused in ampulla of the fallopian tube. So now the embryo has been formed. So after the formation of the embryo, what the process of embryo will be. So now further down, as the embryo is been passing down, it will be forming. It will be forming blastocyst. It will be forming morula, and then further down, after the attachment, it will form the proper fetus, right? So during the formation of the fetus, it has to undergo a step called gastrulation. Okay, so after gastrulation, three main germ layers are formed. These three germ layers you would have heard since the beginning of your 11th, 12th, or even in your school days also. You would have known these three germ cells that will be pluripotent or forms any cells in the body. So those three layers are ectoderm, mesoderm, and endoderm. Okay, so these are the three layers that you will be or you should be able to remember in your exams. Okay, so after the formation, now that we have learned the gametes, the male gamete, the female gamete, they have fused in the ampulla of the fallopian tube. Further down, it has undergone morula. See, when it is passing down, it is going division again and again. Okay, so the cells are being formed, and after attachment in the uterus, it is undergoing something known as gastrulation. Gastrulation is where the fetus finally decides it has to undergo differentiation. Like one has to form brain, one has to form legs, one has to form liver. All these things will be done after the formation of gastrulation. These three layers is what decides what it is gonna be or what the cells it will be forming into. Okay, so during gastrulation, so for your exams, only things that you need to remember is what these specific layers, each layer will be forming into, what the ectoderm will be forming into, what the mesoderm, and what the endoderm will be forming into. So remember, ectoderm is all the outer things that will be formed in the body. Okay, ectoderm is all the outer things that will be forming in the body. Mesoderm is middle. Okay, all the muscles, all the organs, each and everything that will be inside will be formed by mesoderm. Okay, then further down, moving into endoderm, only thing that you need to remember is all the inner linings of, see, for example, the mesoderm has formed gastrointestinal tract, all the muscles, smooth muscles, everything that it has formed. Okay, so inner lining, the endothelial lining or the epithelial lining that will be formed is by endoderm. Okay, so ectoderm, all the skin, eyes, everything will be formed by ectoderm, whereas all the organs, everything will be formed by mesoderm, and whereas the inner lining of the body will be formed by endoderm. So when the examiner asks the question, what is the inner lining of this thing, just if you don't know the answer, just mark for endoderm. Okay, so for the organs and the coverings of the organs, each and everything, you need to go for mesoderm. And when it comes to outer thing, it is gonna be ectoderm. All the nails, skin, eyelids, everything will be formed by ectoderm. Okay, so that is all and everything you need to know here. One more other thing about ectoderm would be the neural crest cells and the neural tube. Neural tube here, except for all the other organs, that is formed by the mesoderm, the brain, the CNS, the PNS, all the nervous system will be formed by ectoderm. So remember it like this. The ectoderm will be the outer thing, including the brain. All the other organs will be mesoderm. Okay. So in the mesoderm also there are three different parts. So that is very, very important, and only those questions that are being asked in the exams. Okay, that I'll also be telling you. But the only thing before going into mesoderm, I want you guys to remember is in ectoderm, the brain and all the nervous system is being formed by the ectoderm. Okay, so that is by neural crest cells and the neural tubes. Neural tube will be forming the CNS, whereas the neural crest cell will be forming the PNS, that is peripheral nervous system. And rest I told you, right? Hair, nails, everything will be formed by ectoderm, the outer layer of the ectoderm. Now further down, let us discuss about mesoderm. Mesoderm has three different things. Okay, one is the central thing. Okay, this is the neural tube which was formed by the ectoderm. Okay, so right next to it there is something known as paraaxial mesoderm. See, axis is there. So next to that is paraaxial, and very lateral to it is lateral plate mesoderm. Between lateral plate and paraaxial there is something known as intermediate mesoderm. Okay, so there are three different things of mesoderm. One is axis is there, which was formed by ectoderm. So right next to it on the two sides it is going to be paraaxial mesoderm, and right next to it on the lateral side it is not right next to it, like very distant to it will be lateral plate mesoderm, and between it will be intermediate mesoderm. So let us see what the three different structures of the mesoderm will form into. Okay. So the paraaxial mesoderm will give rise to muscles and skeleton. See, right next to axis, that is spinal cord, it is gonna be muscles, right? All the muscles of the spine, upper limb, lower limb, all these things will be formed by paraaxial mesoderm. Okay, then towards the lateral side, it is gonna be smooth muscles of the organs. All the smooth muscles will be formed by the lateral plate mesoderm. So between paraaxial and the lateral plate mesoderm, there was something known as intermediate mesoderm. So here the genitourinary system will be formed. All the urinary system will be formed by intermediate mesoderm. This is the only thing that you need to remember in mesoderm. So see, I have made it very easy for you with this diagram, because they will be asking you this exact diagram. Okay, so this was ectoderm which was forming epidermis. This was neural tube which was also formed by ectoderm, whereas these three structures, that is paraaxial mesoderm, intermediate mesoderm and the lateral plate mesoderm, so these three things will be formed by mesoderm, and paraaxial mesoderm will give rise to muscles and skeleton, whereas the intermediate mesoderm will give rise to urogenital system, whereas the lateral plate mesoderm will give rise to urogenital system. Okay, so that is all and everything you need to remember about mesoderm. Moving down, in endoderm, as I told you, all the inner lining, mainly GIT, okay, all the inner lining of all these organs that were formed by mesoderm will be lined by endoderm. Okay, so that is all and everything you need to remember about the three germ layers. Please remember these things. Now the sticky note part, please note down only these three things. Ectoderm will be giving rise to outer epithelium. Mesoderm will be having three different layers, that is paraaxial, intermediate and lateral plate, and please write down the one single organ that these three things of the mesoderm will give rise to, and then the endoderm will be forming the inner lining. So please note down this in your sticky note. Moving down. So one of the major and very important topic of general embryology is the formation of pharyngeal arches. Okay, so there are 6 different pharyngeal arches, and fifth pharyngeal arch is not there. Okay, so that has been dissolved during the formation of the pharyngeal arches. So we will be having 4, that is 1, 2, 3, 4 and 6. The fifth has been dissolved. So there is going to be 5 different pharyngeal arches. Okay, remember it is not 5 but 6th pharyngeal arch. Okay, so each of these pharyngeal arches will give rise to head and neck. Okay, why is it very important? Is because these pharyngeal arches is what is going to be very important in the formation of the head and neck. Okay, head, neck and face will be formed by pharyngeal arches, and there is going to be disorders if there is a fault in this pharyngeal arch formation. That is why the examiners are also keen on asking the formation of these pharyngeal arches. The first pharyngeal arch, see, instead of remembering any of these things, I have told you that there are 5 pharyngeal arches, right? So to the people who have studied a little would be knowing that the cranial you'll be knowing about cranial nerves, that is 5th cranial nerve, 7th cranial nerve, 9th and 10th cranial nerves. These are the important cranials of head, neck and face. Okay, so these 5 nerves will be the ones that will be supplying head, neck and face. You remember, right? So those things is what is gonna save you in exam. 5, 7, 9 and 10. These are the 4 nerves that will be forming the entirety of pharyngeal arches. See, for example, the first arch has a nerve supply of 5th cranial nerve, the second will be 7th cranial nerve, the third will be 9th cranial nerve, the 4th and the 6th will be 10th cranial nerve. So that is all and everything you need to know in the pharyngeal arches. Okay, so 5th cranial nerve, 7, 9 and 10. So remember these 4 nerves and you will be sorted for pharyngeal arches. Okay, because see, 5th nerve, what does it supply? Mastication, right? Chewing. Everything will be supplied by trigeminal nerve, 5th nerve, mastication muscles. Okay, then 7th nerve, facial expression. So facial muscles, facial expression muscles, smiling, angry, everything will be done by facial nerve and facial muscles. Okay, then further going down, 9th cranial nerve, stylopharyngeus, the pharyngeal muscles will be supplied by 9th cranial nerve. Then coming down, the 10th, the vagus nerve, which is the longest nerve in the human body, is the vagus nerve. So this will form the laryngeal and pharyngeal muscles. Remember, the 10th nerve is what gives the supply and the motor supply and the sensory supply to the larynx, right? So that is why it is important. So from beginning, the fifth cranial nerve, that is the trigeminal nerve, will be important in mastication. See, all these things I will be explaining to you when I will be going down to head, neck and face in the cranial nerve chapter. Okay, but for now you remember the pharyngeal arches. There are 5 arches and 5 nerves that I'll have to remember. That is, first will be 5th cranial nerve, 7th will be facial nerve, 3rd will be 9th cranial nerve, then for 4th and 6th pharyngeal arches will be supplied by 10th cranial nerve. Okay, so and the muscles that are formed by these things will be the muscles supplied by these nerves. Okay, just remember in that way and you will be sorted for exams. Then further moving down, it is not only the nerves and muscles that are formed by pharyngeal nerves, the pharyngeal arches, it is also the arteries and the bones that are being formed by the pharyngeal arches. What are the arteries that are formed by the pharyngeal arches? See, deep down, very deep down here, it is gonna be the pulmonary arteries, right? Pulmonary arteries, where does it go? Very deep down, it is gonna be pulmonary artery. It will be going into the 2 lungs. Then moving above, it is gonna be arch of aorta on the left side and the subclavian artery on the right side. So these are the 2 arches. One was pulmonary artery, then it was subclavian artery on the right side and arch of aorta on the left side. Then further moving above, it is going to be common carotid artery on the right side and internal carotid artery on the left side. So further above, we are moving into, see, that is how it is going to be in the formation of pharyngeal arches. Okay, further moving above, it is going to be hyoid and stapedial artery. Then it is going to be maxillary artery. Maxillary artery, see, maxilla was supplied by trigeminal nerve. The mastication muscles were also there, and the maxillary artery is also there. So further down, it is going to be hyoid artery. Okay, then further down it was carotid artery. Then further down it was arch of aorta and subclavian artery, then pulmonary artery. These are the arterial supplies these pharyngeal arches are going to have. Okay, you need to remember pharyngeal arches has 5 arches. It's nerve supply, it's muscles, it's arteries. Now that we have learned these 3 structures, we will go into bone structures that these things will be forming. So 10th nerve, larynx, it is going to be cricoid and arytenoid cartilage. Then again 10th nerve, in the 10th it was, right? 10th will be supplying 4th and 6th. So again it is going to be laryngeal cartilages. Then stylopharyngeus was hyoid, right? Hyoid. Here the stylopharyngeus is there, it will be connecting to hyoid. So that is why in the 3rd pharyngeal arch, you have to remember it as hyoid. Then further moving down, in the facial bones, it is going to be stapes, styloid and hyoid bone. Hyoid will be formed by 3rd pharyngeal arch and also the 2nd pharyngeal arch. Okay, so the stapes, the inner ear, in inner ear there are 3 bones, 3 ossicles, that is malleus, incus and stapes. The stapes is the smallest known bone in the human body, right? So that will be formed in the 2nd pharyngeal arch, and the other 2 bones, the other 2 ossicles of the inner ear, that is malleus and incus, will be formed in the 1st pharyngeal arch. Please remember this, very important. This is very important, because all the other things they won't ask you. They'll be asking you which is exception. Okay, because what we'll be thinking is, okay, ear ossicles, it will be 1st pharyngeal arch, right? Because it is inside ear, it is near the maxilla. Oh, okay, so inside meaning we'll be thinking of 1st pharyngeal arch. But no, it is going to be 2nd pharyngeal arch which will form the stapes. This is very important. Okay, that is all and everything you need to remember in the pharyngeal arches. The 5 different structures there are. They are nerves, muscles, arteries and bones. Okay, so I have given it. So please go through this before going into exam. Okay, once and you will be done for it. Then further down, so in the pharyngeal arches, we know that if you have studied in the 1st year, you would be knowing there are cleft and pouches that will be formed by these pharyngeal arches. So these clefts and pouches will give rise to some of the structures that is important for the head, neck and face. Those things are, in the 1st pharyngeal arch, it is going to be eustachian tube on the inside, just below the 1st pharyngeal arch and the 2nd pharyngeal arch. See inside, imagine. Okay, so just like you are imagining, if you would have seen Good Doctor here, the Sean Murphy will be like he'll be imagining how the human body is like, right? So in the same way, you start imagining the 1st arch is there, below the 2nd arch is there. 1st arch was giving rise to all the muscles and supplies that was formed by 5th cranial nerve and the 5th maxillary stuff. Okay, just below it, so is the facial nerve, the facial part, right? So between that, inside there is eustachian tube which is connected to the ear. So 1st pharyngeal arch and 2nd pharyngeal arch, it is gonna be eustachian tube, remember in that way, and the outside is ear canal. Okay, so eustachian tube and ear canal, external ear canal. Okay, so that is the two things that you will be able to remember. Okay, so never forget these two things. Then further coming down, it is gonna be palatine tonsil. Tonsils will be in the second pharyngeal arch, like right. So after eustachian tube, just below that, it is palatine tonsil. Then just going below down, it will be after the palatine tonsils are here. See, palatine tonsil will be palpating here, right, in the clinicals. So just below down, what is there? Here it's going to be thyroid and parathyroid. Okay, so very important thing is parathyroid. Parathyroid will be in the third pharyngeal arch. Okay, in the third pharyngeal cleft, it is going to be parathyroid. Okay, just below it, within the in between the parathyroid itself, the thymus, which will be degraded after we are growing up. It is a lymphoid organ which will be degrading when we are growing, as we are growing. Okay, so this lymphoid organ will also be in the with the parathyroid gland. Then further going down, it is gonna be thyroid. Okay, so because see, here the parathyroid was formed and here the thyroid is forming. Just remember in that way. So two things, eustachian tube and the ear canal. Further down, what was there? Further down, it was palatine tonsil. After moving down, it was gonna be parathyroid, and with the parathyroid itself, it was thymus. Then further moving down, it was thyroid gland. So finally, all and everything that you need to know about the pharyngeal arches is done. One of the noted points here is, and it has been asked repeatedly in our competitive exams, is the remnant of the fifth pharyngeal pouch. So remnant of the fifth pharyngeal pouch, see, I told you, right, there were 6 arches. Okay, 6 arches, and the fifth arch will be degrading. Okay, for example, if this has not been degraded, then it will form something known as ultimobrachial body. Okay, ultimobrachial body, there is a cyst formation which we will be seeing in your clinicals. I have seen a case of it. So there is a ultimobrachial body which will be formed if there is a remnant of this fifth pharyngeal arch. It was supposed to be degraded, but it was not degraded. That is why the remnant has been formed. So this is the thing that you need to remember. Then on the cleft of the third pharyngeal arch, see, the third pharyngeal arch, I was telling, on the inside it was forming parathyroid, but on the outside, if this cleft remains, then it is going to form something known as cervical sinus. Okay, it will lead to branchial cyst. If the fifth pharyngeal arch is persistent, then it was going to be ultimobrachial body, whereas in the third one, if it is going to be persistent, then it is forming brachial cyst. These are the two things that will go into your sticky notes. Okay, these are the two things that will go into your sticky notes. These two things. Okay, please note down. Then further moving down, we'll be discussing the only 2 topics that are very important in histology. After embryology, general embryology, all you need to know, I have told you. The first thing is chromosome, where the SRY gene is. Then coming to gametogenesis. In oogenesis, the 2 steps of arrest was important. That is, the first arrest will take place in prophase 1, and the second arrest will take place in metaphase 2. Only those 2 things were important. And then further moving down with the formation of germ cell layers, that is 3 germ cell layers, ectoderm, mesoderm and endoderm. So what will these germ layers form into? Only those things are important. Then the 5th topic was pharyngeal arch, and what are the nerves, arteries, muscles and bones that these 5 pharyngeal arch will form into. These are the 5 things that you need to remember in embryology, and embryology is sorted. Okay, so further down, moving into histology, only 2 things that you need to remember, that is histology of cartilages and histology of lymph nodes. Okay, so lymphatic structures or lymphoid organs. Okay, only these two histology has been asked in these many years. Okay, I don't know why they are very keen into asking these things. There is no point in asking this also, but those idiots will be asking these questions itself. Okay, so that is why we'll go and see what are these histology of. Okay, so in hyaline cartilage, I'll be telling you how you can differentiate these things only, okay? Because just before going to exams, you need not remember any of the nonsense I'm gonna tell you. Only these things, because they'll be asking the question of histology. One photograph they'll give you of, for example, hyaline cartilage, and they'll be telling there is a nose cartilage that. The histology of a nose cartilage, and please identify what cartilage is this. Instead of you guys knowing what nose cartilage was forming, just by seeing the histology photograph itself, you will be able to solve this question, right? So that is why the easy way to identify this is, for the hyaline cartilage there will be small cells, okay? So like the ground glass appearance, small cells will be there. Histology of elastic cartilage, you will be able to see the blackening, okay? So the blackening, just like histology, hyaline cartilage itself, you will be able to differentiate it as black and blue. A slide will be given with respect to elastic cartilage, and fibrocartilage will be these fibers, okay? So these fibers you will be able to easily appreciate in the fibrocartilage. Okay, that is all and everything you need to remember in hyaline, fibrocartilage. So this is the entirety of cartilages. So if you wanna know about the examples of what these cartilages are also, just remember hyaline cartilage you will be able to move, okay? These things like only a little bit, nose cartilage, epiglottis, and all these things. Epiglottis is elastic cartilage, nose cartilage, then laryngeal cartilage, trachea, bronchi, all these things are hyaline cartilage. Whereas the elastic cartilage will be epiglottis, external auditory meatus, EEE. Okay, the elastic cartilage is external auditory meatus, then epiglottis. Remember those two things. Then the fibrocartilage will be hard, okay? So in between the intervertebral disc, you know, right, the cartilage will be able to move all these things, but fibrocartilage, the vertebral disc, you wouldn't be able to move, and the pubic symphysis, which is very hard, will also not be able to move, and the sternoclavicular joint, all these things will be having the fibers. So that is how you can remember these cartilages. That is all and everything you need to know about the histology of the cartilages. Further down, we'll have to know, and one more thing is I forgot, was the histology of glands, that is the salivary gland, those secretory glands they are asking. So in salivary gland, so there are three glands, right? Parotid gland, sublingual gland, and submandibular gland. These three glands are in a different structure. That is why they are asking. For example, during the removal, we might remove. Sometimes we would remove sublingual gland or submandibular gland, and we should be able to differentiate, right, as a pathologist. So that is why they'll be asking this, because sometimes there is accidental remove, or sometimes there might be migration of these cells into other glands, right? So that is why it is very important to know. That is how they are asking it. So salivary gland, there are three things, that is parotid gland, sublingual gland, and submandibular gland. In a parotid gland, it is going to be serous acinus. In sublingual, it is going to be mucinous, and submandibular gland, it will be combined or mixed. Okay, parotid gland is serous, sublingual gland is mucinous, and submandibular gland is mixed. Okay, 1, 2, and the combined, this submandibular gland is forming. So serous, you will be able to see in this way, right? There is one single circle and dots of blue are there. Serous is secreting serous salivation, whereas mucinous gland, you will be able to appreciate some flower like pattern. And if both are also there, then it is gonna be, see, both are there, right? Both are there. The flower pattern is also there, then the round with dotted structure is also there. So that is how it is gonna be. So you will be able to differentiate between these serous mucinous and mixed salivary glands. That is all you need to know. Then further down, moving on to lymphoid organs, it is gonna be thymus, spleen, lymph nodes, and bone marrow. So these lymphoid organs is what will form the fighting soldiers of our body. That is antibodies, all the WBCs, all these soldiers will be formed in these lymphoid organs only, right? So these soldiers is what helps fight all the microorganisms inside the body. So that is why it is important to know all these 4 organ histology. So first I'll be telling you how the spleen will be, so that you will be able to differentiate between all the other structures. Spleen, you will be able to see the red pulp, okay? Red, if you see red in the histology, it is definitely spleen, okay? If you see any red color in the histology slide, it is gonna be spleen. That is how you should be able to remember. Don't remember any other thing. Okay, histology, you are not going and writing a first year histology exam. You are just going into a competitive exam. You will be you should be able to remember it within seconds. So that is why if red is there, spleen, red pulp. Okay, it denotes red pulp. That is why spleen. Okay, because the RBC breakdown, everything is happening. That is why spleen is the graveyard of RBC, right? So red cell breakdown is happening. Heme is being secreted from the breakdown. That is why red pulp is there. Okay. Further down, for the thymus, you will be able to appreciate the medulla and the cortex very properly. Okay, medulla and cortex will be separated. That is how you can remember thymus. Red was spleen. Cortex and medulla I am able to appreciate, just like brain, cortex and medulla it is there, then that is thymus. Okay. So then whatever else you see is lymph node. If they are discussing about lymphoid organs, if any of these 2 things are not there, then it is lymph node. Now they have started asking these things also. What is this layer? What is this germinal center? What is medulla and all this paracortex and etc. They're asking. Please note down that too. Just before going into exam, please note down these things of germinal center. If I tell you now, now you wouldn't be able to appreciate this during your exams also. So that is why just before going into your exams, just try to like take a glance of this histology and just know that this is the germinal center and this is the medulla. This is the medulla and this is the paracortex. Okay, in the paracortex itself, you'll be having these lymph node germinal centers, etc. Okay, so see, forget all this. Just remember, if the red pulp is there, red is there, spleen. If the medulla and cortex is differentiated just like brain, remember it is thymus. Rest everything is lymph node. In lymph node, just remember there is one germinal center which is pale and surrounding that is cortex, paracortex. Okay, that is all and everything you need to know. And bone marrow is not much important. Okay, bone marrow is not much important. So this is all and everything you need to know about embryology and histology. Thank you.